On the island of St. Helena, a volcanic speck in the South Atlantic roughly halfway between the coasts of Africa and Brazil, there lives an animal that has outlasted every human being born in the same century. His name is Jonathan. He is a Seychelles giant tortoise, and as of 2026, he is approximately 194 years old. When Jonathan hatched, sometime around 1832, the British Empire was still expanding across the globe. Charles Darwin had not yet sailed to the Galapagos. Abraham Lincoln was a 23-year-old store clerk in Illinois. The photograph had not been invented. Neither had the light bulb, the car, the airplane, or the antibiotic. Jonathan has been alive for all of it. 2 world wars, the invention of penicillin, the moon landing, the birth of the internet. He arrived on St. Helena in 1882 as a gift to the governor, and a photograph taken of him in 1886 shows an animal that was already fully mature, his shell scarred and weathered, his neck thick. He has lived in the same garden, on the same island for 144 years, outliving governor after governor, empire after empire. But Jonathan is not a biological anomaly. He is not some 1 in a billion genetic accident. Galapagos giant tortoises regularly reach 150 years. Aldabra tortoises on the islands of the Indian Ocean approach 200. Adwaita, an Aldabra giant tortoise housed at the Alipore Zoological Gardens in Kolkata, India, reportedly lived to roughly 255 years before dying in 2006. If that estimate is accurate, he hatched before the American Revolution. And this is where the question gets strange, because nothing else their size lives anywhere close to this long. A dairy cow weighs about 700 kilograms, roughly twice the mass of a large Galapagos tortoise. A dairy cow lives about 20 years. A horse of similar weight rarely exceeds 30. A Komodo dragon, another large reptile and an apex predator on its islands, tops out around 30 years. Even among reptiles, tortoises stand alone, outliving everything in their weight class by a factor of 5 to 10. That was the observation that sat in plain sight for centuries. By 2018, the explanation was finally starting to emerge, and it came not from zoologists watching tortoises in the field, but from geneticists reading code inside cells that had been dividing for over a century without breaking down. In biology, there is a pattern so consistent it functions almost like a law. Larger animals live longer than smaller animals. A mouse weighing roughly 20 grams, about the weight of 4 nickels, lives about 2 years. A dog weighing 30 kilograms lives roughly a decade. An elephant at 5,000 kilograms can reach 70 years. A bowhead whale pushing 100,000 kilograms, heavier than a fully loaded semi-truck, has been documented living past 200. The correlation is not perfect, but it is remarkably reliable across mammals, birds, and reptiles alike. More mass, more time. The reasoning behind this is partly metabolic. Smaller animals burn energy faster. Their hearts beat at extraordinary speeds, hundreds of times per minute in a shrew, and their cells divide at a rate that accumulates damage quickly. A mouse heart beats roughly 600 times per minute. Over its 2-year lifespan, that totals approximately 630 million beats. An elephant heart beats about 30 times per minute. Over 70 years, that works out to roughly 1.1 billion beats. The numbers are different, but they land in the same order of magnitude. For most mammals, there seems to be a finite budget of heartbeats, and the speed at which you spend them determines how long you last. A giant tortoise heart beats approximately 6 to 10 times per minute. Over 200 years, at 8 beats per minute, that comes to roughly 840 million heartbeats, well within the mammalian range. So the tortoise is not breaking the heartbeat budget. It is spending it at the slowest rate in the animal kingdom. Every minute that passes inside a tortoise body is biologically shorter than a minute inside yours. The clock is the same. The metabolism reading that clock is not. That observation led to the first serious hypothesis about tortoise longevity. Maybe they just live slowly. Raymond Pearl, a biologist at Johns Hopkins University, proposed the rate of living theory in 1928. The idea was intuitive. If metabolism generates byproducts that damage cells, and those byproducts accumulate over time, then an animal that metabolizes slowly should accumulate damage slowly and therefore live longer. Tortoises, with their cold-blooded physiology and glacial metabolic rates, seemed like the perfect conformation. But the rate of living theory has a problem. Birds. Parrots and cockatoos have metabolic rates comparable to or higher than similarly sized mammals, yet some species live past 80 years. A macaw, burning fuel at a furious rate, outlives a dog 10 times over. If metabolism alone explained lifespan, birds should die young. They do not. Something else was at work. And the clue came from a completely different direction. From a question that, on its surface, had nothing to do with tortoises at all. In the late 1970s, Richard Peto, an epidemiologist at the University of Oxford, noticed something that should have been obvious, but that nobody had formally addressed. If cancer is fundamentally a disease of copying errors, if every time a cell divides there is a small probability that a mutation will occur in a growth-regulating gene, then animals with more cells should develop cancer at dramatically higher rates than animals with fewer cells. A blue whale has roughly 1,000 times more cells than a human. A human has roughly 37 trillion cells compared to a mouse's 10 billion. Over a long lifespan with far more cell divisions, the probability of a cancerous mutation should skyrocket. But it does not. Blue whales do not develop cancer at 1,000 times the human rate. Elephants, with their massive bodies and long lives, have cancer mortality rates of approximately 5%, compared to roughly 11 to 25% in humans, despite having far more cells dividing over a comparable time frame. This contradiction became known as Peto's paradox, and it implied that large, long-lived animals had evolved protective mechanisms that smaller, shorter-lived animals simply had not. The elephant's answer was discovered first. In 2015, Joshua Schiffman at the Huntsman Cancer Institute at the University of Utah, working with Carlo Maili at Arizona State University, published a study in the Journal of the American Medical Association revealing that elephants carry 20 copies of TP53, the tumor suppressor gene often called the guardian of the genome. Humans carry 1 copy. When elephant cells sustain DNA damage, the TP53 response is so overwhelming that the damaged cell does not merely stop dividing. It destroys itself entirely. The rate of programmed cell death in elephant cells exposed to radiation was nearly double that of human cells. Rather than risking a cancerous mutation, the elephant cell simply dies and gets replaced by a fresh one. That discovery opened a new question. If elephants solved Peto's paradox through TP53 duplication, how did tortoises solve it? A Galapagos tortoise weighing 300 kilograms and living 175 years faces the same mathematical problem. More cells, more time, more divisions. More opportunities for something to go catastrophically wrong inside a single cell. And yet cancer in wild tortoise populations is vanishingly rare. The answer arrived in December 2018, when Victor Quesada and Carlos López-Otín at the University of Oviedo in Spain published a landmark study in Nature, Ecology, and Evolution. They had done something that biologists had been waiting years for. They sequenced the complete genome of Lonesome George. Lonesome George was, quite possibly, the most famous tortoise who ever lived. He was the last known individual of the Pinta Island Galapagos tortoise, a subspecies called Chelonoidis abingdonii, that once numbered in the thousands across the volcanic slopes of Pinta Island in the northern Galapagos archipelago. By the time a Hungarian scientist named József Vajgvölgyi spotted him on the island in 1971, he was the only one left. Every other member of his subspecies had been killed, most of them by whalers and sailors, who had harvested Galapagos tortoises for food during the 18th and 19th centuries. For over 40 years, George lived at the Charles Darwin Research Station on Santa Cruz Island in the Galapagos. Conservationists tried repeatedly to breed him with females from closely related subspecies. He never produced viable offspring. On June 24, 2012, his caretaker Fausto Urena found him dead in his enclosure. He was estimated to be over 100 years old. The Pinta Island tortoise went extinct at that moment, in a concrete pen, on an island that was not his own. But George's cells survived. Researchers had collected tissue samples during his lifetime, and after his death, the decision was made to sequence his entire genome. When Quesada and López-Otín's team published the results, the findings forced biologists to rethink what they thought they knew about aging. The Lonesome George genome contained something extraordinary. Giant tortoises carry expanded and duplicated copies of genes involved in DNA damage repair. Where humans have a single copy of certain repair genes, tortoises have multiple versions, each slightly different, creating a redundant system for catching and fixing the copying errors that drive both cancer and aging. The team identified specific gene family expansions related to immune response and inflammation control. They found variants in genes associated with cellular maintenance that had no parallel in any mammalian genome they had examined. The tortoises had not evolved one single trick. They had evolved an entire layered defense system, extra tumor suppressors to kill damaged cells before they could turn cancerous, enhanced DNA repair mechanisms to prevent damage from accumulating in the first place, and refined inflammatory responses to keep the chronic background inflammation that accelerates aging in mammals from ever taking hold. I mean, think about that for a second. A cold-blooded reptile lumbering across a volcanic island at 2 miles per hour had, through hundreds of millions of years of natural selection, assembled a genomic toolkit for fighting aging that surpasses anything the human body possesses. But the genome alone did not answer the deeper question. Knowing that tortoises have these genes is like knowing that a fortress has a fire suppression system. It tells you what is there. It does not tell you why tortoises evolved this level of protection when other animals did not. For that answer, the trail led to an entirely different branch of biology, one that had nothing to do with genes directly and everything to do with who was trying to eat them. In 1952, Peter Medawar, a British immunologist who would later win the Nobel Prize, delivered a lecture at University College London titled An Unsolved Problem of Biology. The problem was aging. Medawar asked a deceptively simple question. If natural selection favors traits that help organisms survive and reproduce, why does aging exist at all? Why would evolution permit bodies to deteriorate? Why not simply select for immortality? His answer was elegant. Natural selection can only act on traits that affect an organism during its reproductive years. Once an animal has reproduced and its offspring are independent, the force of selection on that animal's survival drops dramatically. Mutations that cause harm late in life, after reproduction has already occurred, are essentially invisible to natural selection. They pile up in the genome because there is no selective pressure to clean them out. Aging, Medawar argued, is not an adaptation. It is a failure of selection. A garbage heap of late-acting mutations that nobody is sweeping away. 5 years later, in 1957, evolutionary biologist George Williams at Michigan State University extended Medawar's insight with a concept called antagonistic pleiotropy. Williams proposed that some genes might actively benefit an organism early in life while causing damage later. A gene that promotes rapid bone growth and early reproduction might also promote brittle bones at age 50. Natural selection would still favor this gene because the early benefit outweighs the late cost. By the time the cost arrives, the organism has already passed its genes forward. Together, Medawar and Williams built a framework that made a specific, testable prediction. The rate of aging in any species should be determined by its rate of extrinsic mortality, meaning death from external causes like predation, accidents, or environmental catastrophe, rather than from internal deterioration. The logic is direct. If you are a mouse, and the overwhelming majority of your population is killed by hawks, foxes, and cold weather before reaching 2 years of age, natural selection has no reason to invest in keeping your body functional past age 2. Any gene that repairs your DNA or suppresses your cancer at age 3 is wasted energy because you are statistically dead by then. Better to pour those resources into growing fast, reproducing early, and making as many offspring as possible before something eats you. But if you are a giant tortoise, and you are encased in a shell that almost nothing in your environment can break through, the equation inverts completely. This is where the story takes a turn nobody expected. The turtle shell is one of the most remarkable defensive structures in the history of vertebrate life. It first appeared in the fossil record approximately 220 million years ago during the late Triassic period, when dinosaurs were just beginning their rise to dominance. The earliest known proto-turtle, Odontochelys semitestacea, discovered in 2008 in Guizhou Province, China, by Chun Li of the Chinese Academy of Sciences and published in the journal Nature, already possessed a fully developed plastron, the flat ventral shell, though its upper carapace was not yet complete. Over the following tens of millions of years, the shell developed into a nearly impenetrable fortress. The carapace of a mature giant tortoise is composed of fused rib bones and vertebrae overlaid with keratinous scutes, the same protein that forms human fingernails, creating a layered composite structure that resists crushing, puncturing, and tearing. An adult Galapagos tortoise retreated inside its shell is functionally invulnerable to every natural predator in its environment. Nothing on the islands can bite through it. Nothing can crush it. The only reliable predators of adult giant tortoises across their evolutionary history have been large-jawed crocodilians and, eventually, humans. When Medawar and Williams' framework is applied to this situation, the prediction is almost absurdly clear. An animal that is nearly impossible to kill by external forces should evolve to live as long as biologically feasible. If a tortoise embryo inherits a mutation in a gene that repairs DNA damage, and that gene only matters at age 80, that mutation will still be visible to natural selection. Because a tortoise encased in its shell has an excellent chance of still being alive at 80, the gene will be tested, the mutation will be sorted. Selection will act on it accordingly. Contrast that with a mouse, where a gene that only matters at age 80 is irrelevant because no wild mouse will ever see that birthday. The shell did not just protect tortoises from predators, it fundamentally changed the evolutionary calculus of aging. By lowering extrinsic mortality to near zero, the shell extended the reach of natural selection deep into old age, allowing evolution to fine-tune genes for DNA repair, cancer suppression, immune regulation, and cellular maintenance over hundreds of millions of years. The genome that Quesada and Lopez-Otin found when they sequenced Lonesome George was not some fortunate accident. It was the inevitable product of 220 million years of selection in an organism that almost nothing could kill. The tortoise did not evolve longevity despite its slow metabolism or its simple life. It evolved longevity because of its shell. The armor came first. The anti-aging toolkit followed, assembled gene by gene over geological time because the armor made it worthwhile. The same principle appears everywhere once you know what to look for. Every animal that defies the body size to lifespan expectation shares a version of the same adaptation. Dramatically reduced extrinsic mortality. Bowhead whales in the Arctic Ocean have been documented living past 200 years, verified by amino acid racemization of islands proteins by Jeffrey Batta at the Scripps Institution of Oceanography in the late 1990s. A bowhead whale is so enormous that, once it reaches adulthood, nothing in the ocean hunts it. Its sheer mass is its shell. Greenland sharks, the longest-lived vertebrates on record, can exceed 400 years. In 2016, Julius Nielsen at the University of Copenhagen published radiocarbon dating evidence in Science, showing that one specimen was roughly 392 years old, plus or minus 120 years. These sharks drift through the deep, frigid waters of the North Atlantic at about 1 mile per hour, in temperatures barely above freezing, with essentially no natural predators. Their environment is their shell. Naked mole rats in the underground tunnel systems of East Africa live roughly 30 years, which is extraordinary for a rodent weighing about 35 grams, barely more than an AA battery. A mouse of similar size lives 2 years. The naked mole rat spends its entire life underground, protected from predators, temperature swings, and most pathogens by a labyrinth of tunnels. Rochelle Buffenstein, a biogerontologist who spent decades studying the species at Calico Life Sciences in San Francisco, documented that naked mole rats show negligible senescence and a cancer rate so low that for years researchers believed they might be completely immune. Their tunnel system is their shell. The pattern is consistent across every branch of the animal kingdom. Protection from external death gives evolution both the time and the incentive to invest in internal maintenance. The longer you are likely to survive, the more selection pressure exists to keep your body functional. The investment in DNA repair, tumor suppression, and cellular upkeep that the tortoise genome displays is not generosity on evolution's part. It is accounting. When your body is likely to be around for a century, every gene that prevents deterioration at year 50 or year 100 has a survival payoff. Evolution pays that bill because the bill comes due. If you are finding this useful, subscribe now. Every week, there is another thread in the biological record that conventional textbooks skip entirely. The algorithm stops surfacing this content if you do not subscribe. and the next video connects directly to what you are learning right now. So tortoises evolved longer lifespans because their shells lowered extrinsic mortality and gave natural selection a reason to invest in cellular maintenance. But what Beth Reinke and Fernando Colchero discovered when they published their findings in 2022 was that the extent of this adaptation was far more extreme than anyone had anticipated. In June of that year, a study appeared in the journal Science. Reinke, working at Northeastern Illinois University, and Colchero, based at the University of Southern Denmark, led a team of dozens of researchers from institutions across multiple continents. They assembled the largest dataset ever compiled on aging rates in turtles and tortoises, drawing demographic data from 52 species, including both wild populations and zoo-housed individuals. The results were striking. Roughly 75% of the turtle and tortoise species examined showed what the researchers called negligible senescence. Their risk of dying in any given year did not increase as they aged. In some species, mortality rates actually decreased with age, meaning older individuals were less likely to die in a given year than younger ones. That finding runs directly against one of the most fundamental assumptions in biology. In humans, the probability of dying roughly doubles every 8 years after the age of 30. A 50-year-old is significantly more likely to die in the next year than a 30-year-old. An 80-year-old is dramatically more at risk than a 50-year-old. This pattern, called the Gompertz curve after the 19th century mathematician Benjamin Gompertz who first described it, is so universal across mammals that for decades it was assumed to be an inescapable feature of biological existence. Tortoises simply ignore it. A 100-year-old tortoise is not measurably more fragile than a 50-year-old tortoise. Its cells are not noticeably more damaged. Its organs are not measurably less functional. Its immune system is not detectably weaker. The biological clock that ticks relentlessly inside every mammal, wearing down tissue and accumulating damage decade by decade, appears to either tick far more slowly in tortoises or not tick at all. Reinke and Colchero noted in their findings that protective adaptations like shells reduce environmental threats, contributing to the evolution of slower aging. The correlation in their dataset was sharp. The more protected the species, the flatter its mortality curve. Box turtles, which can seal their shells completely shut with no gap for a predator to exploit, showed some of the lowest aging rates of any species studied. If you are wondering whether that means tortoises are biologically immortal, the answer is complicated. They are not. They still die. Jonathan on St. Helena is going blind and has lost his sense of smell. Old tortoises develop shell rot, succumb to parasitic infections, and occasionally simply stop eating. But they do not die of aging the way a human does, through a progressive, predictable cascade of organ failure driven by accumulated cellular damage. They die of specific events, individual things that go wrong, rather than from a general systemic decline. The system itself does not wear out. Individual components occasionally fail, and there is a real, profound difference between those two ways of dying. The detail that keeps aging researchers awake at night is what this implies at the cellular level. In most mammals, aging is driven in part by the shortening of telomeres, the protective caps on the ends of chromosomes. Every time a cell divides, the telomeres get slightly shorter. When they become critically short, the cell enters a state called senescence, a zombie-like condition where it stops dividing but refuses to die, pumping out inflammatory signals that damage the surrounding tissue. The accumulation of these senescent cells is one of the primary drivers of age-related disease in humans, contributing to everything from arthritis to cardiovascular failure to cognitive decline. Tortoises appear to manage this problem differently. Studies on telomere dynamics and long-lived reptiles, including work by researchers at the Leibniz Institute on Aging in Jena, Germany, have found that some tortoise species maintain stable telomere lengths across their lifespans. Their cells either activate telomerase, the enzyme that rebuilds telomere caps, more effectively than mammalian cells, or they employ alternative lengthening mechanisms that prevent the progressive shortening that triggers senescence in the first place. The result is that tortoise cells can continue dividing, repairing tissue, and maintaining organ function for decades, beyond the point where a mammalian cell of the same type would have entered its zombie state and begun poisoning its neighbors with inflammatory signals. The inflammation picture adds another layer entirely. In humans, aging is accompanied by a condition researchers call inflammaging, a chronic, low-grade inflammatory state driven by the buildup of senescent cells, damaged mitochondria, and overactive immune signaling. This background inflammation accelerates tissue degeneration, promotes tumor growth, and contributes to the vascular damage that drives heart disease and stroke. By age 60, most humans are carrying a burden of chronic inflammation that is measurably degrading their organs every day, whether they feel it or not. The Lonesome George genome analysis revealed that giant tortoises carry specific gene variants associated with tighter regulation of inflammatory pathways. Their immune systems appear to be better calibrated, mounting aggressive responses to actual infections while keeping background inflammation quiet. The slow burn that gradually dismantles a 70-year-old human's cardiovascular system does not appear to develop in a 150-year-old tortoise. The immune system stays precise, the background noise stays low, the machinery keeps running clean. And this is where the biological story collides with a piece of history that had been sitting in plain sight for centuries. During the 17th, 18th, and 19th centuries, Galapagos giant tortoises were harvested by the tens of thousands by whalers, pirates, and naval vessels crossing the Pacific Ocean. The tortoises were not killed on the spot for their meat. They were loaded alive into the dark cargo holds of wooden sailing ships and stacked on their backs in rows. The reason was simple and brutal. A Galapagos tortoise could survive in a ship's hold for up to a year without food or water. Its metabolic rate was so low, its ability to store nutrients so efficient, and its tolerance for deprivation so extreme that it functioned as a living pantry. Sailors could pull a tortoise from the hold 6 months into a Pacific crossing, slaughter it on the deck, and eat fresh meat at a time when every other food source on board had long since spoiled or been consumed. Captain James Colnett of the British Royal Navy noted in 1798 that his ship carried over 100 live tortoises taken from the Galapagos, and the crew consumed them steadily over subsequent months of sailing. Charles Darwin himself, arriving in the Galapagos aboard HMS Beagle in 1835, observed that the tortoises were being hunted so aggressively that certain island populations were already collapsing. Harriet, a tortoise that Darwin may have personally collected during that voyage, eventually ended up at the Australia Zoo in Queensland, where she died in 2006 at around 176 years old. The whaling-era harvest killed an estimated 100,000 to 200,000 Galapagos tortoises over roughly 3 centuries. To put that in perspective, the current total population of Galapagos tortoises across all surviving subspecies is roughly 20,000 to 25,000 animals. The sailors consumed a population perhaps 10 times larger than what exists today. Several subspecies were driven to complete extinction. The Pinta Island subspecies that Lonesome George represented was one of them. The very trait that made tortoises biologically extraordinary, their ability to slow their metabolism to almost nothing and survive indefinitely without food or water, made them perfect targets for exploitation. The same adaptation that allowed them to outlive every other animal in their weight class also made them ideal living food storage for ships that needed provisions to last across open ocean. The irony cuts even deeper when you follow the logic through. The metabolic slowness that keeps their cells from accumulating damage, that keeps their hearts beating at 6 beats per minute, that keeps their telomeres from shortening, also left them almost uniquely defenseless against one specific predator. They cannot outrun anything, they lack any means to fight back, and on an open volcanic island there is simply nowhere to disappear to. Their entire survival strategy depended on a shell that had been effective against every threat evolution had tested against it over 220 million years. Every threat except one, the one predator that hunts by intelligence and technology rather than by teeth and claws. 220 million years of evolutionary investment in longevity, defeated by a sailor with a rope and a cargo hold. The armor that made tortoises biologically near immortal offered no defense against the only species on Earth that could simply flip them over and carry them away. But here is a detail that shifts the entire picture. Despite that catastrophic population collapse, the species survived. Breeding programs launched by the Charles Darwin Foundation and the Galapagos National Park have brought several tortoise subspecies back from near extinction. On Hispaniola Island, a population that had been reduced to just 14 individuals in the 1960s, a group small enough to fit inside a single elevator has been rebuilt through captive breeding to over 2,000 animals as of the latest surveys. And the tortoises released back onto their home islands are not diminished versions of their ancestors. They carry the same genome, the same DNA repair machinery, the same tumor suppression toolkit, the same capacity for negligible senescence that Reinke and Colchero documented. Every tortoise hatching on Hispaniola today inherits the molecular legacy of 220 million years of evolutionary refinement. The genetic code inside their cells is older than flowering plants, older than birds, older than mammals themselves. And it still works, driving the same cellular maintenance programs in 2026 that it drove in the Triassic. The relevance of all this to human medicine has not been lost on aging researchers. In 2013, Cynthia Kenyon, a molecular biologist at the University of California, San Francisco, who had already demonstrated that single-gene mutations could double the lifespan of the roundworm C. elegans, joined Calico Life Sciences, a Google-funded research company dedicated to understanding the biology of aging. One of Calico's driving questions involves understanding why certain organisms age negligibly, while others, including humans, deteriorate on a predictable schedule. The tortoise genome offers specific targets. The expanded DNA repair gene families identified by Quesada and Lopez-Otin represent pathways that could theoretically be enhanced in human cells through gene therapy or pharmaceutical intervention. The cancer suppression mechanisms, the immune calibration that prevents inflammaging, the telomere maintenance strategies. Each of these represents a biological system that functions better inside a tortoise than it does inside a human. Not because the tortoise is more biologically complex, because 220 million years of selection in a low-mortality organism had the time to optimize these systems in a way that 300,000 years of human evolution under relentless predation pressure simply did not. The arithmetic of evolution is blunt. Your body was not built to last because, for most of your species' existence, lasting was not worth the investment. Your ancestors lived in a world where being eaten, starving, or dying of infection before age 40 was the statistical norm. Investing in DNA repair machinery that only pays off at age 80 was, from evolution's ledger, wasted resources. Faster to build a body that reproduces early and dies young than one that endures for centuries and breeds slowly. The tortoise faced the opposite pressure. Inside its shell, every year it survived was another year its genes could be passed forward. And so evolution spent 220 million years, a span roughly 730 times longer than the entire existence of Homo sapiens, making the tortoise's cells as durable as biology allows. What makes this entire story genuinely unsettling is what it reveals about the nature of aging itself. For decades, aging was treated as an inevitable feature of biological existence, as fundamental and inescapable as entropy. The tortoise disproves that assumption in the starkest possible terms. Aging is an evolutionary strategy, not a fixed law of biology. It is a trade-off, a resource allocation decision that natural selection makes differently depending on the survival equation facing each species. Mice age rapidly not because deterioration is inevitable, but because, in a world saturated with predators, investing in longevity yields no return. Tortoises age negligibly not because they are magically exempt from cellular decay, but because, inside their shells, the investment in maintenance pays compound interest across centuries. Both strategies are rational responses to different ecological pressures. Neither is the only option biology permits. The moment you understand that aging is a strategy rather than a sentence, the question shifts. It stops being about why tortoises live so long and becomes something far more uncomfortable. The tools to prevent aging exist. They are inside Lonesome George's genome. They function inside the cells of every tortoise on the Galapagos right now. DNA repair machinery that catches mutations before they become cancer. Inflammatory regulation that prevents the slow burn of tissue damage. Telomere maintenance that keeps cells dividing cleanly for centuries. None of these capabilities are alien to biology. They are proven, field-tested, and running continuously inside living animals today. They just were never selected for in the human lineage, because, until very recently in evolutionary time, no human lived long enough for them to matter. If you had asked a biologist in 1950 whether a 200-year human lifespan was theoretically possible, the answer would have been a flat no. Aging was considered a fixed constraint, like the speed of light. If you ask a biologist that same question today, the answer is more cautious. What stops human cells from lasting centuries turns out to be an investment decision evolution made for us. And investment decisions can, at least in theory, be reversed. Somewhere on St. Helena, in a garden surrounded by old stone walls that have not changed much since the Victorian era, Jonathan is still alive. He cannot see well anymore. He relies on his caretakers to locate his food. But his cells are still dividing. His heart is still beating, roughly 6 times per minute, the same steady rhythm it has maintained for close to 2 centuries. His body is running on molecular machinery that was already ancient before the dinosaurs went extinct. The real question is why you almost certainly will not reach 194 years. Because the answer has everything to do with the fact that your ancestors never grew a shell. If you want to see what other evolutionary trade-offs shaped the world you inherited, click the video on screen now.